Hi everybody. Welcome to our tutorial, our hands-on demo of how to use FragPipe to launch MSFragger database searches. I really felt it important that instead of just talking about the theory of database search engines or how tandem mass spectrometry happens, to actually do a hands-on encounter with the software itself. My goal is that all the students here are able to do this for themselves, that you can take your data that you've collected on the instruments last night and conduct them through the pipeline using MSFragger and friends. I'm aware that MSFragger is one of these really valuable tools for open search. We will not be demonstrating that. We'll just be using it as an ordinary search engine. And the reason why I chose it for today's demo is that when you perform narrow database searches, when you perform kind of the typical kind of protein identification experiment, MSFragger is outstandingly fast. So fast that you can run it on a conventional laptop, even with a fair number of files. So up at the top of the page at the moment, you can see that I'm connected to the web server at nesvilab.org. Alexei Nesvizhky has been a force in the field of proteome informatics for his entire career since he was a postdoc, and he's been a real leader in the field of how to do protein identification well. Probably the tool that most people would know him for would be peptide profit and protein profit, which are part of the transproteomic pipeline. Very, very widely used tool. Even if you've never used peptide profit, protein profit, you've probably used some software along the way that is built on that same model. So very influential. Okay, so MSFragger is the search engine we want to run. But I'd like to take just a moment to look through the files that I've contributed by thumb drive to everybody in here. So you should have received a directory that looks something like this. We have a directory of installers, the MSFragger 2.1 execution directory. You have this file that's got an accession on it, the MSV blah, 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 blah. That is a massive accession number for a particular set of raw files. We'll talk about that in just a minute. And you have the Philosopher software unzipped in a directory ready to go. We also have a FASTA file here called FAS. We have a jar file. This represents, I think many people have heard of the Java programming language. Frequently, when you bundle up a piece of software that you've completed writing in Java, you package it up as a jar file. So instead of giving people a huge spray of files, you can just give them one, and it's executable. So the FragPipe is the user interface created by Nesvilab for launching MSFragger, as well as downstream tools, which are called Philosopher here. And finally, we have a README file that I've created. Now, the README is pretty straightforward. It points out that the demonstration that we're working with is based on one that was written by Sarah Haynes, who is a researcher in the Nesvizhky lab. You can see her original write-up at that URL. I point out that the data files we're working with came from the iPRG 2009 test. This was a test created by a group that included me. And the experiments themselves were run at Vanderbilt University back in 2008. And we have a sequence database that we're working with, and this describes the exact commands that I used to prepare that for use. So as usual, the README file just kind of gives you some hints about where all this stuff came from. I mentioned that there's a tutorial online created by Sarah and friends. You can follow that here. This is what you would get if you followed that URL. You can see they've got pretty good screenshots and explanations about what each of these steps is useful for. I believe they even have raw data that you can download to run on exactly the same files as they use there. We are going to diverge from this map quite a bit, so we'll just sort of follow the path I've laid out today, but you can always do this one to refresh your skills in a slightly different context. Okay, so if we go through the directories, we see that we have a bunch of installers. Now, you didn't have to install any of this, hopefully, to make everything work on your machine. But the IDPicker part that we're going to run later will require that you have that software installed. Otherwise, you can just follow on screen. That's not a problem. You can see that we have a file called JRE here. This is a Java runtime environment, which is to say that if your computer is not already set up to run Java software, for example, to be able to execute jar files, you would need to install that JRE in order to be able to do that. I would further mention that I've included the 64-bit Java virtual machine here. If you are using a machine that has more than 4 gigs of RAM, you definitely want MSFragger to have access to all of it, because it can do really, really great things to speed up code by using lots and lots of memory. So if your machine has more than 4 gigs of RAM, you should definitely install the 64-bit version of Java in order to benefit from that. Now, I must say a legal word about MSFragger 2.1. MSFragger is not software that anybody just gets to download and do whatever they want with. I can't, for example, download MSFragger, slap it on a CD, and sell it to you. That would be illegal, right? This is software that's available under license. We were given permission in this limited case for me to distribute it to you. If you are using MSFragger all the time, please go to the University of Michigan website, follow the prompts, and actually sign the license agreement to continue doing that. It doesn't cost you anything because you're academics, but it is appropriate that you use a licensed version, not just one that some random bozo on the street gave you, right? So the other thing is that the software is always being updated. It's under very active development by the mzVishki group, and you will find that if you watch it for even 6 months, you won't be on version 2.1 anymore. You'll be on version 2.3 or 2.6. They're incredibly fast in their development right now. Now, Philosopher is, as I mentioned, tools, a set of tools that take place downstream, mostly, of MSFragger. We don't just identify spectra in a raw file and then say we're done. We need to be able to decide which of those spectra have been successfully identified, which ones we would actually trust. And we also need to infer a set of proteins that explain the peptides we've observed. These tools in this framework, the peptide profit and protein profit tools are used for those purposes. And here, rather than installing the TPP itself, which is rather big and ungainly, we can install just the Philosopher tool. And if that's installed on your machine, you're good to go. It has all of the things you really need to make sense of your data. Plus, it's, again, under very, very active development. It's very much worth your time to follow on Twitter the people in the mzVishki lab who are working on this, so that you get a sense of all the cool things that they're adding. So I noted that IDPicker is some software that we'll be using at the end for interpreting the database identifications that we produce. But this other tool that I've included is called ProteoWizard. Now, ProteoWizard is a very widely used tool in the space of proteomics, which is very gratifying because we had a little part in making it. But if in most cases, when you are analyzing proteomics data sets, you're not going to be able to do it from raw files. This is one of the advantages that MSFragger has. It can work directly from raw files. So we don't have to use ProteoWizard today, but if you are going to analyze these data by another platform, you're probably going to need ProteoWizard in order to move them into a format that you can use, such as mzML or MGF, the mascot generic file format. Okay, so that's just a word about the installers. We'll come back to some of these things later on. I mentioned MSFragger and Philosopher have both been unzipped so that we can just use them. But I also want to take a moment to spend a moment with our data set itself, the MSV. So this was created, as I mentioned, for a comparative test. The Proteome Informatics Research Group of the Association for Biomolecular Resource Facilities, nice long name I know, was very interested in determining whether people would find the same proteomic differences between two samples. Now, that's a very unlikely proposition. If you give people a bunch of yellow E. coli and a bunch of red E. coli, and you ask them which proteins change, you're going to get tremendous analytical differences because some people will use long gradients and some people will use short gradients, some will use gels, some will use all kinds of things. But in this study, we decided to control for all of that by giving everybody the same set of 10 raw files. So we have 5 quintuplicates of red E. coli and quintuplicates of yellow E. coli. And by giving everyone exactly the same raw data and giving everyone exactly the same sequence database, we thought we would remove a lot of the variability in which proteins people would call differential between red and yellow E. coli. It's a really fun experiment. In any case, the data are really, really clean. There's very little technical variability in them. And we also have an answer key that we developed out of what we call the blue and green sets, but we won't go into all of that at the moment. But I would point out that all of these raw data are publicly available at the MSV number that appears up here at the top. That is at the massive repository at the University of California, San Diego. So you can see that there's a bit of metadata that's visible right there in the raw file names. You see that these first 5 are all called red, and these last 5 are all called yellow. So we've given you a hint in the file names just how these things split into 2 cohorts. Right, so I would also point out that these data collected on an old school Orbitrap XL, I think it's been a while since I looked at it, are not as big as some of the files that you would get from a contemporary mass spec. If you bought if you bought yourself a Fusion and analyzed these same samples, you would probably find that your files were much more than even a gigabyte in size. But you can see that each of these is clocking in at about 1/3 of a gigabyte. This is the reason why I had to give you 4 gigabytes of files in order to work with today, because the experiments produce large reams of data. All right, now to go ahead and get us kicked off, I think it would be really valuable if we start the FragPipe software. So FragPipe 11.0 jar is the software I want to run. Because I have Java installed on this machine, I can just double-click it or hit enter on it, and it will open. That's really handy. Oh, I actually have 2 copies of it open now. Ha. So one of the things that's happening here, I'm going to close my other copy in the background here. There we go. One of the things that should happen immediately when you launch FragPipe is that it's going to sniff your computer, like a little dog. But the goal here is that FragPipe knows that it itself doesn't do a whole lot of interesting stuff. I mean no insults to the people who created it. This is designed as a GUI. It's a wrapper. So the software is making it easy for us to use MSFragger. It's making it easy for us to use Philosopher. But the tools that it's calling are not part of FragPipe itself. It's just calling those externally. What that implies is that the software must locate your copy of MSFragger and your copy of Philosopher when it runs. In this case, that's been very straightforward because this machine was already set up. But normally, I might have to browse to find that. So if I started in the CPGR tutorial directory, this is the set of files I gave you, I would need to specify that MSFragger jar is found inside the MSFragger 2.1 directory and MSFragger 2.1 jar. Now, that might be a little confusing, right? The software interface says it's looking for msfragger.jar, but I've located msfragger-2.1.jar. So the software is going to have to validate whether this file I've pointed it to really is an MSFragger, and we see that there's no problem there. It recognizes it just fine. Now, I want to point out a gentle word about bioinformatics researchers. Bioinformatics researchers live and die by the cred we get for people using our software at the end of the day. If I am the world's greatest bioinformaticist, but I'm the only person who can make use of my tools, I'm not going to be the world's greatest bioinformaticist for long, right? So in the same way, we see that this user interface specifies that we should cite that article when we use it. So if you're writing a paper in which you use the Sequest engine, or you use the FragPipe engine, or you use MSGF+, it is polite to cite the paper that introduced that search engine. This should be common sense, but just the same, it's worth saying. I would also note that the software FragPipe is a really useful interface for deciding whether or not you are up to date. Because when you tell it to go to MSFragger 2.1, it's going out to the network and sniffing to see if there's a more recent version of the software there. So even if you haven't been watching the Twitter feed, your user interface here might say, hey, you really need to think about updating your code. So very handy. Now, Philosopher is a little different. Now, we noted that MSFragger was a jar file, but Philosopher is an exe. This is an executable program. The jar file will run whether you're running on a Mac or Linux or on a Windows PC. This exe down here, Philosopher, will not do the same. If you're not running one of the platforms supported by Philosopher, you're not going to be able to use that. In this case, we see that the path to it is inside that directory that we copied from the machines. It's right here in that directory. Okay, so at the moment, we only care about these top two, MSFragger and Philosopher. Python down at the bottom is useful for some of the advanced features that are available to you in Philosopher within the FragPipe user interface, but in this case, we will not be using it. So we don't care if you don't have Python installed on your machine. I would note that the same YouTube channel that you're looking at right now, hello, my online viewers, you will find that there's a series of videos from earlier this year that talk about how to use Python and how to develop Python tools online. So, great. So, with that said, we now have all of our configuration in place. That is a great place to be. So, let us now move on to the next tab, Select LC-MS-MS Files. Naturally, if you're going to run a search, you're going to need to specify where to find those data files. If we were using something other than MSFragger or, say, Mirror Match, we would probably need to convert these files to another format. So I'm going to take just a moment to look at what that looks like. So I would start with, I'm going to move to my program files. If you have ProteoWizard installed on your machine, you can follow along doing that as well. But in this case, we are going to use the msConvertGUI software. So this is a .NET wrapper around the msConvert command line tool. Okay, so I'm going to just start msConvertGUI. As you see, I have this user interface that lets me import a set of raw files and output a different file format. So in this case, I could specify that I want to go into my CPGR tutorial, go into the MSV directory, and I could grab one or all of these raw files at this point. I'm going to pick one of them, specify that I'm going to open that one, and add this one to the conversion list, so this one will be processed. I want to write an mzML file in this case, and I want to use zlib compression. But there's one other thing I want to do. Very frequently, when people are getting their data ready for analysis in a database search engine, they don't want to have peak shapes, they just want individual peak lists. So this is a tool that allows us to do this. We can specify that we want to use peak picking. I want to use the libraries made available by Thermo for that purpose, and anything that's either an MS or an MS scan, I want to filter in this way. Then I add that filter. At that point, I could click Start, and the software would dutifully read the raw file and write out an mzML file. I would point out that it's often quite a lot easier to just remember the settings you want and create a batch file to run it on the command line instead, but a lot of people feel much more comfortable in the confines of a graphical user interface, so it's important to have that capability. So ProteoWizard is often thought of as the way you get mzMLs. It actually has a lot of other tools built into it, but we're not going to fuss with that right now. But I want you to be aware that if somebody says, can you give me these data in mzML format, there's an easy and free way to do that. ProteoWizard is here for you. Okay, I'm going to close down ProteoWizard now. So in this case, MSFragger has gained the ability to read raw files directly. So I'm going to specify that I want to add files from this directory. So again, if we have the directory that we copied from the thumb drives, we see that in the massive folder, we had 10 raw files. I can just say Control A for select all to grab all of them, and then say select. Now,
mine says found problems with
yours is contains spaces. That's very interesting. Oh, that's interesting. Okay, so you've run into actually a pretty common problem with software. Very frequently, people write their code in such a way that if there's a space in the path to the file in one of the directories that leads to it, it loses the capability of remembering the whole path together. So if you move these files to a location where there's not a space in the path, that will solve that problem. But I will be sure to mention that to the developers, and they'll catch that problem. In my case, you see I've copied the files into CPGR-tutorial, but there's no space in there. If I'd made this CPGR space tutorial, I would have hit the same problem you did. So yeah, that's what seems to be causing the problem there. I want to note that the yeah, you'll need to probably, since you're moving both the raw files and the jar, you might, well, it might work. It might work. Okay, good. Well, great. Good news. All right, so at the moment, we know more about the raw files than MSFragger does, or than FragPipe does. You remember that our file names told us that some of these represent reds and some of these represent yellows. We are going to use the FragPipe interface to tell us how many spectra are seen for each protein for each input file, right? Because we're prepping for a comparative experiment. We want to know which proteins change between red and yellow. So we need to tell the user interface more about the data it's about to process. And if I double click right here, well, I always struggle to get this thing to allow me to edit. Oh, there we go. If you double click it just right, you should be able to edit that field. I'm going to call this one red 1. I'm going to double click on the second one and call that red 2, red 3. Oh, I made a typo, but that's okay. Come back and fix that. Red 4. All right. If you're an X-wing pilot, what do you call Mark? What do you call Mark Hamill in his X-wing? Luke Skywalker flies what? He's red 5. All right. Sorry, we have a question, a more relevant question. Oh, okay. Okay. All right, so this is going to be yellow 1, yellow 2, yellow 3, yellow 4, and yellow 5. This is going to make it a whole lot simpler to figure out which samples are contributing identification data for a particular protein. Okay, great. So we've done all of that. Next, we need to go to database. And here I'm going to take a brief moment to tell you a backstory. I'm going to show it right here. Philosopher is, let us say, a little bit particular about how you get your sequence databases. To begin with, I had downloaded a database from Ensemble Bacteria for E. coli, because all of these samples represent E. coli K12. But I found that I ran into some problems with Philosopher when I was doing that. So in this case, I have used I actually read the manual, I have to say. I followed the directions on how to download a sequence database designed for use with Philosopher. So on the command line, and this is the part that I know may just cause a lot of people to quiver and shake, and I'm sorry about that. I have used a Philosopher workspace. I create the Philosopher workspace so that I'm allowed to use Philosopher functions. I've then used the database command within Philosopher to download a SwissProt, a UniProt full proteome for E. coli. And I want to note that, I've given it an ID number here. Where on earth did that come from? Well, I started over at the Uniprot website. When you're at Uniprot website, you can specify that you want to search for a particular taxon, and once you've done all that, you are likely to get to a place where you can find a reference proteome for that species. You want to look for this kind of the red file folder with the dog-eared corner with the R on it. That means that you've got a carefully annotated, long-studied model organism proteome to work with. That's going to be great if you're working with E. coli or yeast or mouse or whatever. It's not going to be so viable if you're working with lobster. Although who knows, maybe the lobster people have caught up by now. So you can see that this reference proteome has the proteome number UP blah 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 625. In this case, on the command line, I've told it that I'm interested in grabbing that proteome and I want to add contaminant sequences. Contaminant sequences in this case means that even though we ran an E. coli sample, there might be human proteins in it, right? I mean, maybe I didn't have my gloves on at all moments, or I had dandruff or something awful like that, and a little bit of human protein got into the mix. So I'm going to add those contaminant sequences so I can identify contaminant proteins in the mix. But I want to point out that the software is able to download for me from Uniprot that proteome database. It fetches the database online, and within 30 seconds, I guess, I had a set of sequences downloaded from there. We are going to use what's called a target-decoy method for evaluating our where the false matches are and where the true matches are. And so we needed to add decoy sequences to it. The software does this for us. It is able to create then a unified database that has all the contaminants in it with all of those proteins in both ordinary orientation and in reversed orientation form, and writes that to our directory. So we can see that this file right here was created through that process. I believe, if you're a lot cleverer than me, that you can simply compel the software to download for you this database. But the directions for how to do this in the command line are shown right here. You initiate a workspace, you specify that you want the contaminants added to a particular ID proteome, and it'll create it for you. In this case, they specified that they wanted to use reviewed. Now I'm going to ask if this is obvious to everyone in the room what they meant by that. If you're using a Uniprot database and you specify reviewed, what does that affect?
Doesn't that mean that somebody else looked at it?
It does. It does, yes. Uniprot, and particularly Swissprot, is distinctive in that it has a large group of human curators that examine sequences and confirm whether they're legit or not. In effect, what this means that every protein in Uniprot knowledge base comes from either the reviewed side of the shop or the automated side of the shop. A sequence that hasn't yet been evaluated by a human curator is usually called part of the Tremble database, TREMBL, translation of EMBL. It's only after a human curator has looked at that sequence that it becomes part of Swissprot, the human reviewed side. So in this case, their directions specify that you should use the reviewed flag, which means that it would only grab the proteins that had passed human curation. Now, I think that's a really bad default behavior. I wanted to grab the whole proteome, the whole reference proteome, and that's why I grabbed that set. So I left reviewed out. In this case, I need to specify where to find the FASTA file. It's in the root directory of the stuff that you copied from the thumb drive. So you should have a .fas file here. I would just point out that FASTA files can be a little idiosyncratic, that some people will call them whatever.fasta. Some people will call them whatever.fa, like Ensemble does. Some people will call them FNA if they're nucleotide sequences, and other people will use FAA for amino acid sequences. In this case, they're using yet another one, FAS, rather than FASTA. These things happen, we just try to roll with it. Okay, and you also note that it's telling me how many entries we've got. 9,012 entries. So I'm going to ask you a nasty question. How many proteins are there in E. coli?
Like 10,000.
She says 10,000. I'm going to play over and under. Everyone is going to be forced to make a call on this one. Is Savannah's 10,000 proteins too high or too low? Too low. Someone else said too low. Too high, too high. We have a mixed judgment here. All right, I'm going to pop back over to E. coli. What do you see? She's too high. There are, in fact, about 4,400 proteins in E. coli. If you look at something more complex, like the eukaryotic yeast, that's about 6,200 genes. And humans probably get by on something like 70 to 100,000, but that's allowing for a lot of isoforms, transcript differences, stuff like that. We only have about 20,000 coding genes in humans, protein coding genes. Okay, so how do we get from 4,391 to 9,012? There are no isoforms in bacteria, so that one isn't true.
Is affecting the contaminants?
Right. Very good. So in downloading this database, we used the contaminants command. So it added on probably more than 100 proteins known to be contaminants in common proteomics experiments. So that's one thing that happened. But there's another thing that happened in that we created decoy sequences. For every normal protein sequence in the database, now probably around 4,500, we have created a reversed copy and added that to our database. So that means we've doubled the size of the database in order to be able to evaluate our error levels. Okay, great. If I'm going too slowly, if I'm belaboring points that are just obvious to everyone, I can go faster, but I wanted to make sure everyone followed this. All right. For our first attempt, we are going to use just MSFragger and not the downstream tools. Okay? So I'm going to start by specifying that I want to load defaults for closed search. The software will just sort of leave itself in the last way you had it configured. So if you're trying to recreate a state, it's best to just reload the defaults at the start. It then asks, do we want to update the options for the other tools? Yes, we do. Okay. So there are a lot of things that we can configure here, and I'm not really going to obsess over every last thing here. MSFragger is capable of some very advanced searching techniques. I mean, it's one of the first tools to really make it easy to do open search, but we're not trying to do anything that complex here. What we want is a good protein inventory, and we just want to do this search. So I would note that the software has a sensible set of defaults if the data you're working on came from a QExactive. They're great for that. But the data we're working with are older. Remember that we did this on an Orbitrap, and the yellow and red E. coli were measured in an Orbitrap where the tandem mass spectra were actually low-resolution scans. So we can't run this configuration just as it is. We definitely need to change the fragment mass tolerance. We need to specify that instead of having parts per million fragment mass tolerance, we actually had an M/Z kind of tolerance. 0.5 is what we're going to use. So I'm going to explain that we have a mixture of high-resolution MS scans and low-resolution tandem MS scans in this file. This type of data collection was really, really common, well, in 2008 and 2009. It's not so frequently what you see done today. A lot more people today use HCD than was true back then. So if you're working with a data set that's got some history on it, like this one, you're probably going to need to allow for the fact that it has much more accurate measurements of precursor ions, of peptides, than it does of fragment ions, which here are, each fragment ion is only known to be accurate within 0.5 M/Z. It was measured in an ion trap. Okay, so this up top precursor mass tolerance looks really, really high 50 ppm. I mean, when we measure things in high-resolution mass spectrometry, you expect to have an error of no greater than 10 parts per million. But I'm going to point out that the downstream software is going to take advantage of these mass differences to help it determine which spectra are identified correctly or incorrectly. So we're not going to change that. We're going to leave it as is. We are, however, going to alter this little feature, this calibrate masses thing. You can see that the software is capable of multi-pass searching, where it does one cut at the data and then tries to figure out how accurate the precursor masses really are and then rerun the search. We are not going to use that because we're running on our laptops, and that's a little pokey and slow. So, I mean, it takes twice as long to do the search. So instead, we're going to turn off calibrate masses. We can see that the software is, however, allowing for isotope error. It's possible that the mass spec makes an error in telling us which one is the monoisotope out of the isotope packet seen for a particular peptide. We're going to just leave that in place. Yes?
Can this be run on the server?
Can this? Yes, absolutely.
On the server backend?
Yes.
Okay.
Yes. If you're running on a Linux server, if you have a desktop computer, if you have a monitor attached to the computer, you can, I think, even run FragPipe as your user interface. However, a more typical way to use a Linux server is headless, without a monitor attached. So for something like that, you would probably want to run MSFragger on the command line. The very good news is that the FragPipe user interface can export the configuration file that you then use on a command line computer. So it can save you a lot of time and headaches on that. Okay, we note that we specify what kind of enzyme is being used. Those defaults are all correct. In most cases, the set of modifications is reasonable. Why would we look for a plus 16 on methionine? It's the top line of modifications there. Why would we look for methionine to gain 16 mass units? Water is 18, so we're not hydrating the methionines. What's 16? Hydrogen is 1. Oxygen. Oxygen is a mass of 16. Okay, so we would expect that frequently methionine, being a twitchy little thing, if you give it enough time on your bench, it's going to oxidize. It's going to oxidize. So instead of having its normal mass, it's going to be higher by 16 mass units. In almost every database search that people use in the wild, you will see an oxidation of methionine configured. What about this next one? We have a mass shift of 42 on a square bracket caret. Well, helpfully, the tooltip gives us a hint on that. What is a square bracket caret? We see in this little list that a left square bracket is a modifier for the protein N-terminus. Okay, so so why would we look for an N-terminal change of 42 mass units on a protein? Cell biology is feeling a little old and crusty today. That's all right. Well, very frequently we see that the N-terminus of a protein is acetylated. Acetylation has a mass of 42, and so the MSFragger defaults will always look for a mass shift of 42 at the N-terminus. All right. There's one other change I really want to note down here, and these are things that show up very, very frequently, so I want everybody to know about them. You see that we have enabled a mass change on cysteine, but it is a fixed modification. So this is different than what we had on acetylation of N-termini and oxidation of methionine. We've specified that this is a fixed modification, which means anytime it sees a cysteine, it's not going to assume cysteine has its ordinary mass. Instead, it's going to add 57 mass units to it. And something you did yesterday pertains to that. What would cause our cysteines to be 57 mass units higher?
Maybe the alkylation and reduction.
Very nice, alkylation and reduction. Great. Yes, so you probably used dithiothreatol to break disulfides. Then you treated the mix with iodoacetamide. Iodoacetamide adds this alkylation, a carbamylation specifically, to the side chain of cysteine. That is a mass change of 57. So every time the software encounters a cysteine in the search, it's going to, instead of treating it as 103 Daltons, it's going to treat it as 160 Daltons, a 57 mass unit change. There are other things down here. We're not going to pay attention to them at the moment. Instead, we're going to move ahead with our search. Now, as I said in this very first pass, we are not going to run peptide profit and protein profit. I'm going to diverge from the online tutorial because we're going to use IDPicker to look at them first. So I'm going to run nothing on downstream. I've unchecked peptide profit. I unchecked protein profit. Crystal C should already have been unchecked, I think. We need to tell the software what kind of reporting we want it to do. In this case, we are only using FragPipe to launch the search. So we don't need it to create a report in this particular case. I'm going to uncheck Create Report. Finally, I come to the Run tab, and this is the place where we tell it where we need to write its output. So I'm going to try to browse for a convenient place to put this. It's not got a default value here, so I need to point all the way back to my C drive. Hello, C drive. And I'm going to specify somewhere in CPGR tutorial. I'm going to create a directory called result search only, because this is just running it on MSFragger. Okay. So I'm going to select the result search only directory that I just created, and that's where it's going to write it. Now, for this next piece, we want to have what we call a dry run in bioinformatics. We want to do we want it to show us what the list of commands will be that are generated from this, because we're going to run 10 database searches. So when I click Print Commands, I now have this long list of things. We see that red 1 through 5, yellow 1 through 5, each map to these individual raw files, and it's going to launch 11 commands. Yes, sir?
Sorry, I missed that last part. You check dry run and then you just run.
Oh, I actually didn't check the dry run. This is called a dry run, but I'm just clicking Print Commands, which actually will do a little bit of bug checking to make sure you have a sensible configuration. So that's a handy feature. We see that it's going to execute 11 commands, and it's going to launch MSFragger with this command. It's going to launch the second one, the third one, the fourth one, and then at the end, that's it. It's a pretty short list of things it needs to do for this case. So without further ado, I'm simply going to click Run, and hopefully magic happens. So we see up at the top, there are all those different configuration options. You see those blah, blah, blah equals 0, blah, blah, blah equals minus 1.5, etc. Those are the configuration information that the software is going to use. By running MSFragger in this way, it's making a report of how it was configured when you do that. Let me tell you, you do not want to lose that file, because the time is going to come where you want to publish your work. And when you publish your work, you are required by the journal to tell them how you conducted your search. And that information is all in that configuration file. There's a puzzle happening, so I'm going to check in on how those are going.
I'm not able to run the list.
Okay, is it complaining about the space in the
Yeah, memory.
in the path name?
It just says here, insufficient memory, predicted minimum memory is 4 weeks.
Ah, all right. So you've run into a catch, a nasty little cache that shows up with MSFragger. MSFragger is really, really fast, in part because it is able to use lots of memory to do these things quickly. In your case, your machine has run out of RAM available to it in order to make that search happen in the space you need. So I'm really sorry on that one. Are other people experiencing the same thing? You had a different error.
There's not enough space, so it's, I don't know.
Not enough space on your hard drive?
No, I have enough space on the hard drive. Well, it's not that much. I don't know why it's expecting.
Look here. Let me see what kind of error it's throwing.
Error, that could not. Is that
Ooh. Okay. You may be using a 32-bit Java virtual machine. even if you have more than 4 gigs of RAM on your laptop, if you run with a 32-bit virtual machine, it can't see more than 4 gigs of RAM. So that could be the problem. So you have a laptop with 4 gigs of RAM and a 64-bit VM, but it's saying, I need more memory than 4 gigs to run this search. See, I didn't run this demo on a 4 gig machine to test that, so I'm really glad to have that observation. My laptop has 8 gigs of RAM, and that's just enough extra to make the difference, I think. I should have caught that beforehand. I'm sorry about that.
I just can't install the actual Java GUI.
Okay, and is it behaving itself on your machine?
I got 32
Ah, good, good. So it is running on yours. How much RAM have you got?
8
8 gigs of RAM, okay. Well, now I have learned that if I'm doing this demo in the future, we need to have a configuration that's a little bit more memory friendly. All right, now I understand that brain work is hungry work, so I'm going to make sure that these custard-filled buns get back over here. You're not going to eat one? Oh my goodness. Okay, there you go. This one has lots of icing. Who wants that one? Okay, I'll eat it. Teacher's privilege. Okay, great. So things are happening behind me on screen, and it's worth exploring what that entails. The first one, I'm going to just roll backwards, so I'm going to move the thumbtack back a little bit. I mentioned that we had a whole bunch of configuration information. Up here at the top, you can see that the configuration information is being reported up at the top. Where to go? Is this my dry run that we're looking at? Okay. FragPipe configuration is given here. MSFragger is getting to work. Here's the main search. You see the parameters. Oh, if I let go of the thumbtack, it goes away. Sorry about that. Oh, all right. So parameters. This section is showing how the search engine is going to be configured. Then there's this indexing action. See the number of unique peptides thing of length 7, 8, 9, and so on. I want to explain that at some length. So in the old days, most search engines had no memory of which peptides had already been compared to tandem mass spectra. So if you had the same protein sequence showing up in your database twice, it would digest it twice and compare those peptides to spectra twice. It's not very efficient. One of the most recent an innovation several years back that has now percolated into a lot of other search engines is that the software is able to digest up the entire protein sequence database at the start, sort all of the peptide sequences, and make sure that each one gets compared only once to the tandem mass spectra. So creating an index of the sequence database is one of the first steps you see when you're using a sequence database for the first time with a search. And you can see that it limited itself to peptides of length 50. Now I'm going to do a special thing while this is running, where I'm going to start the task manager in the background and look at performance. You see that I'm sorting my tasks by how much CPU they're using. And right now, BatMass, for a moment there, BatMass was taking up all of the CPU, and it wasn't doing anything else. Now, Java is using all of the CPU. That was for the very last run, too, so I barely caught that in time. So it's worth trying to analyze that. I would point out that in this case, MSFragger is not working from mzML files, and that means it needs to read raw files. But it doesn't know the format of raw files. It is only able to access that through a library made available by Thermo. And that library is called BatMass. So in effect, MSFragger is saying, hey, BatMass, can you help me with this raw file? I don't speak raw file, but you do. You translate this to Spectra for me. So BatMass does this translation from raw to a data structure that MSFragger can read, and then MSFragger does the analysis. So actually what just happened is that, first off, we just searched 10 raw files in the space of 5 minutes on a laptop. That would not have been possible a couple years ago. One of the things that is most shocking about that 5 minutes is that much of that time was spent reading files. Remember that each of these files is about 1/3 of a gigabyte in size. And even if you're working off of an SSD, like this machine is, you still have some amount of time required to read all of those bytes. Then they had to be interpreted by the library, and then and only then could it compare all the peptide sequences to spectra. If we were to repeat this search with something like Mirror-Match, a search engine I wrote several years back, our software can read raw files through the same libraries that theirs can. And yet, the time required for a fully triptych search in Mirror-Match is at least an order of magnitude longer than this. We would not have been able to execute that in 5 minutes. So the speed that has become available to us through open search algorithms like MSFragger is quite astonishing. So you might say to yourself, what good was it? What did we accomplish by running this search? And for that, I'm going to come back to our results search only. Okay, so you remember that we had read 1 through 5 labels that we put on things. I mentioned that we had a configuration file and so on, and a configuration for FragPipe itself, and what we have is a log where the software tells us all the things it did along the way. So this result is pretty clean, really. If we look in red 1, we find a pepXML file. Now, from our pre-demonstration chat, I hope you all remember what pepXML files represent.
Peptide spectrum matches.
Very good reference to notes there. Yes, peptide spectrum matches. Good. So a raw file is the collection of spectra themselves. And here it's exporting a pepXML file that represents its best guess about what each of those spectra represents. If I open up my... Emacs is a text editor that only a geek could love, so I'm not suggesting you install this. But one of the good things it can do is read very large files. You see that this one is 12.5 megabytes. But that's not going to cause it to shrink like a violet, so this is good. So we see that we have an XML file. Probably none of you have ever looked at one, and you would feel much more comfortable if you continued in that blissful state. But XML is a very useful way to store piles and piles of information. So we look, for example, at a spectrum query. Here we have a particular tandem mass spectrum that we've searched, but we're not storing the spectrum here. We're storing its hypothesis about what that spectrum represents. We see that the search hit for FGALA-LTIR, this is a triptych peptide derived from the sequence database, had a mass difference of 0.91 blah, 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 and it had these scores associated with it, hyperscore, next score, expect. So this is the software's evaluation of how much confidence it has that this spectrum is indeed a good match for this spectrum. This spectrum is a good match for this sequence. That's what the XML looks like underneath the scenes. Obviously, it's very, very painful if you, as a proteomics user, are asked to dig through this to figure out which ones you trust and which ones you don't. It's simply not a very intuitive activity. So we have created some software called IDPicker that makes it really easy to interact with your data graphically. So if you've already installed IDPicker, you could just start it by clicking on its icon. It should take just a moment, and you see that we have a user interface that so far doesn't look very descriptive, right? It's just got a file menu, and that's actually the place where we want to start. I'm going to import these results for the search. So if I import the files, I go to the C drive, to the CPGR tutorial, and now I want to go into my result search only directory. And I need to specify that I want to search subfolders, because you remember those pepXMLs are not in the result directory. They're in one directory down, in red 1 and red 2 and red 3. So I'm going to tell it to search the subfolders of our result search only directory. I hit this button, and all of those are added to the list. And you can see from a quick glance all of these different files, these different pepXMLs that it located. Yes, sir?
The result directory did you create?
It was created when FragPipe ran,
Okay, because I ran FragPipe
and it crashed.
and it worked now.
Oh, it works now. That's good. Okay, where did you tell it to save the results?
Oh,
Oh, okay. You found the results?
Yeah, no, it's fine. I'm being stupid.
Oh, no worries. Maybe I think its default location is My Documents.
No. I saved it in a file called run within the actual, you just run in here.
Okay. Great. Well, if you can find them, that's wonderful. All right. So I'm telling it that I want to process these 10 input files as one big experiment. Okay. Now from here, I just click Open, and the software begins sampling those. Oh, wait, it's asking me an important question. We are reading 10 input pepXML files, each representing the PSMs from these 10 raw files. We want to create one assembly that spans all of them. I'm going to just use its default location to save all those results. All right. So it's located these 10 files, and something kind of curious has happened here, and I want to explain why it's really okay. I've told the software I want to sniff these 10 pepXML files and read those IDs in. But the software says that it has found X-tandem search results. That's very weird, isn't it? We ran MSFragger, but it thinks it's got X-tandem results. The answer for why that's happened is that MSFragger reports scores that are the same kinds of scores as what X-tandem reports. Reports a hyperscore, an expectation value score, stuff like that. So the software is going to think it's reading X-tandem results when it's really got MSFragger results. And in this case, that doesn't matter at all. What does matter, however, is this decoy prefix. Remember that we have created decoy sequences in our database so that every sequence is matched by a reversed one. But it's not called R-, it's called rev underscore. If you don't have that right, the software is not going to get anywhere. These other settings don't matter as much. I always change this one to 1 and this one to 0.05. Which is to say that I only care about top-ranked hits. I only care about the best peptide hit for each spectrum. And this means that I'm not willing to consider any peptide that's got a more dubious false discovery rate than 0.05. With that, I tap the OK button. You can see that the software is now going to slurp up each of these 10 input files and create one assembly that spans all of it. Happily, that goes pretty quickly. So in this case, we can wait maybe a minute or so, and we will have an assembly that includes all of the underlying input data. Yes, there's a question.
So the decoy prefix was rev underscore, was that?
REV underscore, yes.
Just to make sure.
Yep.
Max rank and max FDR, sorry, I was navigating to this point.
Oh.
Okay, so max rank is 1 and...
Max rank is 1, FDR I used was 0.05, yes. All right, if you're doing proteogenomics, this prompt matters. If you're not, you can simply skip it. So I would skip this step. All right. So that part hopefully did not seem too threatening. It's intended to be easy to use software. And we spent a lot of time working to make it so, but I appreciate that it may still seem a little foreign to us. So I'm going to try to walk through this bit by bit. At the moment, we have a protein view up at the top. We have a spectrum view, and we have a filter history down here. It should seem relatively clear that the protein view is giving us information about which proteins we hit. Some of it is pretty clear. We had, for example, 362 spectra identified for the DNA-directed RNA polymerase. You would kind of expect that that's one of these genes that you have a lot of in a given cell. We see the accession over here at the left that represents the entry in the SwissProt database that represents it. We have some things like cluster and count that are not going to be that apparent, I think, at this stage. But we do have this coverage plot. And I want to note that sequence coverage is something people often care about quite a lot when they're trying to understand the biology of a particular protein. If, for example, you're interested in whether phosphorylation at the C-terminus of a protein affects its behavior, and you didn't identify any peptides at the C-terminus of the protein, that's going to matter to you. So this coverage orange image is actually an image from N-terminal to C-terminal of the protein, rating how deeply covered different parts of that protein are. So it's quite a lot of information right there. In total, we saw 61 different peptide sequences for this protein. For those 61 peptides, we saw some of them in multiple oxidation states or in different charge states. And so that's recorded by the number of distinct matches we saw. Of course, we did 10 replicate shots of E. coli here. So in a lot of cases, we're going to have captured the same peptide in the same charge state, in the same oxidation state, multiple times. Because we had, I mean, even if you saw that match just once in a given raw file, you could still see it 10 times if that peptide showed up every time. So we can see then that the number of filtered spectra seen for a protein shows up here. Now you might ask, were all 10 of these files giving us similar amounts of information back? And for that, we can click on this little drop down and get information back about each of the groups that were assigned. In this case, the software remembered that we read all of these pepXMLs from different subdirectories, and so it has a memory of how these are organized. I'm going to show, so I asked the question, are all 10 of these files giving us the same amount of information back? So here's red 1, here's red 5. Do we see that all 5 reds have similar numbers of spectra identified successfully? This really could be a little larger and a little bit more in focus. I'm very sorry about that. In this case, the low number we saw was 3464, and the high number we saw was 3718. So do we think of that as an acceptable range, or is that just wild and crazy gyration in the mass spec? Should it identify exactly the same number of spectra every time? We have a yes, we have a no. We have a no. The mass spec has kind of what seems like a relatively random process for deciding which peptides get fragmented every time. It's using usually a top 8 or top 15 kind of model where it says which ions produce the highest intensities in the MS scans, produce tandem mass spectra of those, unless I've seen them recently. That's the algorithm. So which spectra get captured in a given mass spec run is quite random, seemingly so. It's at least complex and chaotic. So it stands to reason then that there's going to be some amount of variation in how many spectra get identified in a given run. We can look at at cohort changes as well and say, did we have more spectra observed typically in yellow runs than in red runs? In this case, the variation is pretty reasonable. This is these technical replicates have very little noise in them. In real-world experiments, seeing swings in identification rates of as much as 25% is quite commonplace. I want you to think about how reproducible you are at the bench, right? Are you a robot? You are not a robot. You know, are you measuring exactly 20 microliters this time and every time? No, it was 19 this time and 21 the next, and so on. Human variation and mass spec variation together can lead to some rather large swings in what identification sensitivity is across these experiments. Before we completely leave behind this result, I want to talk a little bit about why IDPicker is going to be useful to us. And this mostly swings to what we call a pivot table. In this case, we want to be able to track how many spectra were seen for each protein across these 10 groups that we have. So I'm going to click that box and then click somewhere else. Always thought that was a little counterintuitive. And now, as you see, as I scroll to the right, I have this useful kind of information that's come back. Now, the names are a little counterintuitive here. See, result search only, blah, 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 blah. I'm actually going to fix that by a file that I included for you already. So along with the raw files, I gave you one bonus one. If I pop back into the massive directory, you should see a file there called assemble.txt. You can see that I've given each of the files a friendly name. I've got a red cohort and then a replicate 1, 2, 3, 4, and 5 within that, and a yellow cohort, and within that replicates 1, 2, 3, 4, and 5. This file is designed for informing IDPicker how these files relate to each other. So it could just use the names of the directories, but they're kind of long. So instead, I'm going to come back to IDPicker and use source grouping and specify that I want to use that assembly file to label each of them. Here we go, assemble text. So now we see that instead of using the directory names, it's using our friendlier names. Okay, so I want to redo the pivot tables, this time using our friendly names. Uncheck that. And that stuff goes away. Redo the pivot options, spectra by group. These names just are going to work a lot better. Okay. So now, if I'm a statistician trying to say which proteins changed between red and yellow, I can look at, I could look at the overall numbers, right? Here I see there were 184 spectra seen in red for this protein. There were 178 seen in yellow. Who would vote that that's a significant change? Ideally, what would you like a significant change to look like? A whole bunch of counts over here and nothing over there, right? Okay, so that's an extreme case. But I mean, everyone would agree if you only saw it in red, and you always saw it in red, and you never saw it in yellow, that's a change you can really get behind. All right, well, there are some of those in here, but generally speaking, we're given a lot more ambiguous case when we try to analyze these. So let us look for a moment at this protein. This is GROW-L. Okay, so we see that there were 319 spectra seen for it in red. There were 101 spectra seen for it in yellow. Yes, sir?
I'm very much behind what filters are used in databases and such.
I use the standard settings.
Okay.
I use defaults here.
Okay, no, that's fine. I don't know what happened in Eclipse. I'm getting completely different data. Yeah, just okay.
I'm not sure why that is happening, but we can go through that together. We will do that at a later date, I'm afraid. So, all right. So in a case like this, we might have a more compelling difference. There are 3 times as many spectra seen for this protein in red than in yellow. You could, in a very simplistic interpretation, say there's 3 times as much of this protein in red as there is in yellow. A reviewer 3 is going to slap you down for being facile like that. At this point, though, we might be able to make the case that there are different amounts of this protein in red and yellow, with more in red than in yellow. That is a safer statement, okay? But what's a statistician going to say to you about significant differences? It's not just a difference in the means. You also must have the variance under control. So let us see not just whether this holds up in all of red versus all of yellow, but in the subruns. So we have 61, 68, 62, 63, 65 measurements of tandem mass spectra in each of those first 5 red files for this protein. And over here, we have 16, 21, 19, 25, 20. Do we have high or low variance within those?
Relatively low.
I would say that that's pretty low variance. It does bounce around a fair bit, but that's to be expected because you just have a lot of chaotic collection of spectra here. So a protein like this, you can make a pretty compelling case, is different between red E. coli and yellow E. coli in this experiment. Okay, so a lot of the times when you're doing a biological experiment in proteomics, you're not just seeing what's there. You can do that. There are times when that kind of experiment is appropriate. Generally, people are doing proteomics because they're going to compare cohorts. And for that, spectral count tables like this are your bread and butter. This software, of course, is quite friendly in letting you dump off that result to R or even to your friend Excel. And you can simply open that table in Excel using the little export button in the upper right corner. You can see that after a moment, Excel is over here blinking saying, I've got this table for you. And there's your data. So we hope that the software like IDPicker makes this a little easier to work with. Now in the next example, we're going to use we're going to return to FragPipe, and we're going to look at how you would do this using Philosopher not exporting off to using IDPicker. Because once you've installed FragPipe, Philosopher, and MSFragger, you have everything you need to create tables like this in that software instead. So for now, we're going to sign off from this demo and take a little break, because I think we're all yawning a little bit. At least my eyes are running. And we'll come back here in a minute to come back for looking at doing the same operation, this time handing off MSFragger results to Philosopher.